So while I'm trying to get the AV connected here, I want to thank everyone, ILSI and the NIH especially, for organizing. Despite me being tapped for 2 topics this morning. So as was mentioned, I'll go through 2 sort of halves of one topic, I should say. Giving a quick summary of biological topics and challenges in defining a healthy gut microbiome, and then technical concepts and challenges after that. And I think the first half will probably be a little longer than the second half, but I'll try not to delay the break too much. So, if I'm lucky enough that this works. No, okay, we'll do it the old-fashioned way. Let me try one more thing. So one of the things I was struck by in preparing for this talk is that the concept of a healthy microbiome has at least penetrated the public consciousness at this point enough to find pictures when you do a Google image search for it. But sort of to pick up where Claire left off, defining a healthy microbiome has been a challenge for sort of two major areas. One being that, contrary to sort of very early expectations, it's very difficult to find features, be they microbes or otherwise, that are really core or highly prevalent, even among fairly heterogeneous healthy populations. And even when looking at different microbial community configuration types, Claire mentioned the enterotype concept, it's difficult to find uniquely highly populated regions of microbiome configurations. There's much more of a continuum of many different microbiome configuration types that can arise even in a healthy population. And if you squint really closely at this example of a collection of different microbiome configurations, in this example, healthy and diseased microbiomes are overlapping in their configuration space. So I would say that these 2 early assumptions of what would be an easy way to define a healthy microbiome have been the big pieces that didn't work out as hoped, and so we've been left looking for more complicated, more challenging ways to define health. And something that hasn't been mentioned yet that I think is important to keep in mind is a more ecological perspective, especially when comparing microbial ecology to sort of macro scale ecology, because we all have intuitions from day to day of walking through a forest. And it's no surprise, for example, when being in sort of a healthy forest, there's no one common thing. There's no particular tree that you can find in every healthy forest, even though there are some typical trees in a particular region, for example, that you might find. Similarly, not every healthy forest is the same. It doesn't even occur in the same kind of environment. And we're not surprised by this based on our day-to-day experience. We should keep these sort of ecological properties in mind when wrestling with the challenge of a very small ecology that we can't see as intuitively, like the gut microbiome. So I'll use a couple of examples today from the Human Microbiome Project, and Claire mentioned this briefly in passing, just to give a very, very quick context of what these populations and what these data will be. I'll draw from both phases of the Human Microbiome Project, the HMP1 that was published a few years ago and focused on the combination of demonstration projects and the healthy cohort that the consortium worked around. And then I'll draw also from the more recent HMP2 that ran a series of 3 complementary projects focusing on more dynamic functional properties of the microbiome in disease. And I won't go into the details of either phase of the HMP today, but again, just some quick vital statistics so you know what data you're seeing when I grab an example. The HMP1 was published in 2 different waves. The original publication about 5 or 6 years ago now, and a recent update that I'll refer to as the HMP1-2 that added more data to the same population, which again briefly comprised 300, quote, healthy subjects clinically screened, adults, men and women. And I'll draw mostly from the HMP1-2 data set, which includes 2,400 shotgun metagenomes from across the body, including the gut, at up to 3 time points spanning roughly 1 year. So vital statistics on HMP1, 300 subjects, slightly longitudinal, and some examples both from the gut and from outside of the gut using shotgun metagenomics. The HMP2, our particular project, focused on inflammatory bowel disease, which includes Crohn's disease and ulcerative colitis, in case you see these abbreviations on the slides, in addition to control subjects, about 130 that were sampled very longitudinally, biweekly for roughly 1 year each, resulting in about 1,600 total shotgun metagenomes and a bunch of other multiomics that I won't really show today, but there are metatranscriptomes and metabolomes and whatnot that I'll mention occasionally. So these will be the two main populations that I draw examples from when talking through a couple of points that I think both define what we do and don't know so far about a healthy gut microbiome and give examples of what some of the challenges are. The big one, of course, being that there are no particular bugs that are shared among even a relatively homogeneous population like the HMP1, and other simple summary statistics like alpha diversity tend not to really capture very much of the whole picture. Much of this arises from the huge inter-individual variation that I think many people in the room are already familiar with. Even while our genetics might be 99.9% plus identical, even in the fairly homogeneous healthy cohort of the HMP1, two individuals in the gut were lucky to share more than about 10 to 20% of their microbial species. So when somebody says tremendous inter-individual variation in the microbiome, this is the kind of change that we're talking about even within just a Western healthy adult population. There are, on the other hand, many typical microbes, and these are the common trees from my previous slide. There are prevalent microbes that are typical of a particular environment, be it the gut or elsewhere on the body. And these can be thought of as one of the markers of sort of healthy ranges that Claire mentioned, or other simple summaries like alpha diversity, the caveat being that there can be very wide ranges of these simplest summary statistics, even in a healthy population. So as we look across the body, or in the gut, for example, if we think of alpha diversity, the number and types of different organisms in a community, as one of the simplest possible metrics, even in the healthy adult population of the HMP1, there's a huge range of that one summary statistic, let alone comparable statistics like which bugs were there. One of the things that provides the flip side to this coin, the first sort of optimistic piece here, is that while absolute measures of health seem to be pretty challenging with these types of summary statistics, relative measures and changes over time within subjects start to become fairly informative. So to draw an example from the HMP2, which was so intensely longitudinal over time, if we look at baseline differences in a summary statistic like alpha diversity across the control Crohn's disease and ulcerative colitis populations, there's really not much of a difference. But when we look at dynamic changes, when an individual suddenly drops in even a very basic summary statistic like alpha diversity, those tend to correspond with periods of more active inflammation, be it in Crohn's or in ulcerative colitis. And this corresponds with one of the ecological concepts that you'll see applied to microbiome health in terms of movement around this sort of configuration space that I mentioned a few slides ago. That if you think of overall microbial community configuration, not in terms of individual features, but as moving between sort of healthy or unhealthy states, the ability to stay within a particular configuration or be resistant to change, and not to move out of whatever your home state is into a less healthy or dysbiotic state, is a way to think about ecological health more dynamically within the subject over time. There are other inter-individual and microbiome changes or differences that make health difficult to define. An easy one that we won't really need to talk about here are the big differences between even very similar microbial ecologies. So we'll be confined mostly to the gut today, but something to keep in mind is that the gut microbiome is highly distinct as far as human-associated microbial communities go. And to draw one example from the oral cavity, the microbial communities at even very closely physically located microbial niches can start to differ surprisingly ecologically. Within the gut, this is true as you look across, for example, microbes in the lumen versus at the colonic mucosa versus in the small bowel. These are small differences relative to huge differences that you might see body-wide. but they're still substantial relative to those couple of percent variation that we mentioned earlier that can correspond with external factors affecting the microbiome. And then we've already mentioned a few times that the tremendous inter-individual variability, both between these different microbial ecologies and even within a single environment such as the gut. So again, looking at these types of beta diversity changes in a relative dynamic sense rather than an absolute fixed sense can start to be a little more informative. So if we draw a quick example from the HMP2 again and ask how much change we see not between individuals but within individual over time, the baseline is quite low, and that's true both in, quote, health and during inflammatory bowel disease, with some exceptions. So if you look along the top here, this y-axis just summarizes within subject change over time in the gut microbiome. And you'll see that all of the outliers tend to occur during IBD, in our IBD subjects. And these are the ones that occur during those periods of more active inflammation, more active disease that I mentioned a slide or two ago. So this is another way in which we can dynamically capture movement around these microbiome configurations through this sort of state space of the microbiome. I do want to emphasize that the time period over which the microbiome integrates information starts to get really interesting. I find it scientifically really interesting because it's in between a lot of the time periods that we think about for transcriptional regulation, which can happen very, very quickly, versus genetic regulation, which can happen very, very slowly. The microbiome can effectively integrate information over a period of years to decades, this really interesting intermediate health-relevant time period. And something that I think is also kind of conflated with geography and race and ethnicity, as Claire brought up earlier, are early life exposures. So we can see effects where differences in very early life exposures in the microbiome, here drawing examples quickly from a third different cohort that was recruited across 3 different sites in Scandinavia. Again, sort of ignoring the context here, we're looking at population averages across 3 geographical locations in the first 3 to 4 years of life. And here we see effects of geography and location essentially immediately after birth, even at a gross population average scale, where in Finland and Estonia, we see one early life microbiome configuration that rapidly stabilizes. In the Russian cohort with very similar genetics, very similar demographics, we see a different early life microbiome configuration that continues to change dynamically for those first couple of years. And these early life exposures can be locked in some sense, both immunologically and in terms of microbiome composition itself for up to years or in some cases decades. So one of the things I've always found most surprising about the HMP1 is that if you look at this cohort of healthy 20 to 30-year-old adults for the most part, one of the pieces of information we have about HMP1 participants is whether they were breastfed as an infant. And because of the population size, we get a small but statistically significant enrichment, even at the whole phylum level, for but just between the entire Bacteroidetes to Firmicutes ratio in infants that were, in adults that were breastfed as infants. And so this is now several decades later, we can still see echoes that were locked into the microbiome, presumably by these differences in early life exposures. And this is a much smaller difference than something like this. This has already come up previously, but most of what I've spoken to so far has been based on microbial taxonomy or microbiome composition. And there are important contrasts when thinking about microbiome composition taxonomically in terms of who's there versus functionally in terms of what they're doing. And a slide that I've used for years that I think calls this out well is the difference between taxonomic composition in the healthy gut microbiome of the HMP1 and functional composition in the metagenomes of the same subjects, summarized either to the phylum level or to the high pathway level here. And so I need to clarify whenever I use this slide that there are important functional differences between individuals despite this greater level of consistency, just like the small differences in our genetics are functional and very important. But not just in the gut, but throughout the human microbiome, there's a much greater degree of functional consistency than there is of compositional consistency in microbial ecologies. And so this speaks to what Claire mentioned as far as starting to think of healthy ranges of microbial function that might be easier to define than healthy microbial compositions. The problem, as I'll get to later, being that these are, to date, much less well studied than these. One way to link taxonomy and function that I think has really just emerged in the conversation over the past couple of years is when considering microbiome composition at the very strain-specific level. If you talk about, for example, E. coli being part of the microbiome, my E. coli, even when two of us share E. coli as part of our gut microbiome, my E. coli doesn't necessarily have the same genetic composition as yours. This is easy to forget in light of human genetic similarity, but microbes of the same species can differ by a third or more of their gene content. Not SNP level variants, but whole gene level variants. So for folks used to human genetics, that's easy to forget. But if we think of a microbial strain as a taxon that is composed of a specific functional makeup that carries around specific bags of genes, we can start to relate who's there to what they're doing. So this is a quick example where if we look at a series of different E. coli strains across many different samples, metagenomes and isolate strains, and we unroll their genomes to ask which genes are either present in red or absent in yellow in different strains. There's a little block that was highlighted over here that were drawn from gut metagenomes in Nick Lohmann's study of the German E. coli outbreak several years ago now. And so there are groups of genes that define not just E. coli generally, but the strains of E. coli specifically that were in these outbreak-associated metagenomes. And these groups of genes link this taxon to functions that are directly responsible for these E. coli being pathogenic, these E. coli being more adherent, invasive, and toxin-producing, specifically if you squint real hard over there. And this is in contrast to the bulk of genes that make up, in this case, the E. coli core genome that spans all of these strains. But I think this type of strain-specific functional definition has really only become metagenomically accessible in the past couple of years and starts to link this highly diverse taxonomic perspective with a more consistent but less well-characterized functional perspective on the microbiome. Particularly because when you zoom way in and look at individual strains, persistent carriage of specific strains within an individual is part of what defines those long-term health effects or microbiome ecological effects that I mentioned earlier. So if we compare which strains of a particular organism are carried by different individuals, there tend to be, as I mentioned earlier, huge genetic differences between those strains, even when they're of the same, nominally the same organism. Within a subject over time, and this is measuring up to about 1 year each within either the Human Microbiome Project or the MetaHit study, call it 6 months to a year each. There tend to be no or very few changes in which strain of a particular organism is carried. And other studies have taken this out to several years, say 3 to 5 years with confidence at this point. There's a probability of acquiring a different strain or a new mutation sweeping in, but for the most part, microbial strains within a subject are stable for months to years. The problem then being that we have these nice strain markers now that define properties of an individual's microbiome, that link taxonomy and function, but the genes that make up that function are often not yet nearly as well understood as the taxa that make up the compositional analysis of a microbiome. And that's true even in the well-studied human gut. If we take the HMP1 population again and look across individuals at the gene families that we encounter, about a quarter of them tend to be well annotated. We have a good understanding of what their potential biochemistry is. Roughly a quarter of them tend to be highly novel, don't have substantial sequence homology to anything that's in a reference database. And the remaining 50% or so of genes are somewhere in between. They might have homology to a sequence that we've seen before, but without necessarily good biochemical annotation. This is the situation in the gut. Again, we're thinking mostly about the gut today, but I want to call out that every place else is even more challenging. There's a huge amount of arguably health-relevant strain composition that's not functionally characterized, that's not biochemically characterized. So to wrap up the first half here, which I think has indeed gone a little over half, there's greater commonality in functional ecology than in composition of ecology with respect to health, especially as we saw earlier among populations. I haven't really called that out here. It's poorly captured by most simple summary statistics. I think one way or another we're going to be stuck with a fairly nuanced definition of microbiome health. But we can now start to link composition and function in the form of really zoomed in strain-specific microbial definitions, which tend to differ among individuals, but are stable within individuals over time, which really gives us hope of understanding how they contribute to an individual's health phenotype. And so some ideas based on this about how to start defining a healthy microbiome might be dynamic departure over time from a normal range, as Claire called out earlier. And this could be defined through some combination of a within self, a self-defined normal range, and a population or a population-wide normal range. The former requiring a fair degree of quantification. This is perhaps easier to analyze at this point, but harder to generate technically. The latter, being population-wide definitions, is similar to what we saw earlier from Barbara's discussion of, say, nutrition recommendations, or, say, sports drug testing uses the same type of population-defined standards. So for the first chunk, I want to pause there.